Yeah, so final talk of the day, our one keynote of the conference. It's a real pleasure to introduce Dr. Tait Burkhard. Christine has been here for 10 years-ish, I would say. She did her PhD in Utrecht on coronaviruses. I tried to recruit her as a postdoc, but she turned me down and took a postdoc with Alan Archibald here instead. Yeah, and it was the right decision because it was a 3-year project to make a transgenic pig that would be resistant to porcine reproductive respiratory virus. And she did that very successfully. That pig is now licensed to genus and will hopefully be on the market in the near future, maybe. She then took up a tenure-track research fellowship in the Institute as her own group on PRRS, but shortly after that, the pandemic hit. Christine was the only proper coronavirologist in the university. She was also the only person with a functioning CL3 in the university. So the pandemic was a very interesting time for her. She contributed an awful lot to the university's response. Not always getting the credit she deserved, I think.
Did you take it, Paul?
You know, one of the pleasures of being an academic is the friendships you build up over the decades. Anyway, Christine successfully passed her tenure review and she's now a full group leader in the Institute. As you can see, she's also incubating a child, and she's doing this talk 2 weeks before her due date. She's basically Superwoman. So with that, I'll shut up. Christine, over to you.
I'll try my best, yeah. Okay, brilliant. Let's try and work technology now. For the last time today, it's been pretty good. So we're going back to the alpha coronaviruses where we started today, and we actually had a lot of cat talks today, and it's not only due to this outbreak that we're going to talk about now. What we have termed feline coronavirus 23, the emergence and spread of a feline infectious peritonitis occurrence due to a pathogenic canine feline recombinant that is prone to in-host deletion of part of its spike domain. So let's take a step back and actually look back at the feline coronaviruses, because we have had a lot of discussion about this today. And feline coronaviruses are actually a very interesting cluster of one species, alpha coronavirus 1, that encompasses 3 different hosts. It encompasses cats, dogs, and pigs. All of these viruses seem to have evolved from one canine coronavirus, canine ancestral coronavirus, initially giving rise to serotype 1 coronaviruses in cats and dogs, and then further recombining into different serotypes, giving rise to the serotype 2s in cats and dogs, and from there, likely from the canine serotype 2, jumping to the pig to induce transmissible gastroenteritis virus, and further mutating with the deletion that we're going to find back today into porcine respiratory coronavirus, which was a saving grace because it's a natural vaccine, basically, for transmissible gastroenteritis virus. But this is a beautiful example of the complexity of coronaviruses, the recombinations that they can do. They're far more than one virus in this species, even though it's one species. And we've also heard about this. How does feline coronavirus come from a benign feline enteric coronavirus to a very pathogenic feline infectious peritonitis virus? There's various theories. One of them is the internal mutation theory. Another one is the virulent-avirulent theory, which says that both virulent and avirulent strains are circulating and you just luck what infects you. But the internal mutation theory, with a spelling error, not mine, is actually that something triggers a change in the virus that leads to a different tropism of the infection that is far more prone to infect macrophages and spread systemically induces cytokine storm, similar to what we see in human, and that causes then the systemic disease of feline infectious peritonitis. It is, however, contrast to SARS-CoV-2, inversely correlated with age. So most of the feline infectious peritonitis we actually see in juvenile cats under the age of 1 in a normal case. But in 2023, we saw a massive occurrence of feline infectious peritonitis in Cyprus. Cyprus is the renowned island of cats. There is an estimated 1 to 1.5 million cats on the island. And we have seen some very scary numbers. One of the papers quoted 300,000 dead cats due to feline infectious peritonitis. The problem is it's very difficult to quantify that. We have official reports from vets which estimate the case rate to about 12,000 cats that died so far. However, if we're talking to the shelters, they reckon that they have lost between 30 and 40% of their cats in that intense phase in the middle of 2023. And that's where the extrapolation of 300,000 came from. The reality is going to be somewhere in between. But it is a large number, and it's a big question as to why this has happened. Cat density, genetics, and the virus are all potential contributing factors here. This is how the outbreak started, and the estimate is that it started in the divided capital city of Nicosia. There's actually a hypothesis that it actually had already been going on in the northern Turkish part of the city for a while, as in kind of end of 2022. But it's very difficult to validate that because far less testing is done. And there's also a big issue with toxoplasmosis in the Turkish part. So they have other things to worry about. But it spread very rapidly to the different areas, the different provinces of the island. And let me just get the pointer here. You can see that we had a big first wave, which is also represented here, far easier to read, in the first half of 2023, and you could first expect, well, we have lots of kittens in the early year, but the outbreak kept on happening. We're still far above the previous levels of FIP cases that have been detected, and it keeps occurring. We've actually gone higher again in July 2024, so we keep having cases of this outbreak of feline infectious peritonitis. And what we see here are only RT-qPCR confirmed FIP cases, both clinically and RT-qPCR confirmed. What is very strange, though, that our average age of cat is far higher than what we normally see when we have feline infectious peritonitis infections, which already indicates that at least pre-existing immunity doesn't necessarily protect from this infection, because feline coronavirus was present on the island. So the question is, what is happening? The other thing that is very strange is that we see a very high percentage of neurological cases, roughly twice as many as we normally see. FIP can present as kind of the normal wet version, where the cat gets a distended abdomen, which you can see here, jaundice, and a systemic infection. But neurological and dry can present with quite different symptoms. We could also be looking at a potential diagnostic bias, because knowledge about the neurological symptoms is better. More vets are actually diagnosing neurological cases, but it's still a very distinct change to how the different forms are distributed. We also see a reasonably strong staining for FIP in the colon, which indicates that a lot of virus can get shed through the orofecal route, which then would also make reinfections of the next cats potentially very dangerous. The first problem that you always have when you work with companion animal samples, and people have hinted at this today, is that you're working with the offcasts from diagnostic samples. Targetedly sampling companion animals requires a lot of ethical hoops, and it's far easier to work with diagnostic offcasts, but you have very little sample as a consequence. So we actually decided to go for an Oxford nanopore tiled amplicon approach to eventually sequence this virus. Of course, we didn't quite know what we were expecting in the first place. So this is the short version where everything works beautifully in the end. It took us a while to get there, but we now have over 20 full genome sequences from this virus and over 150 partial sequences for these viruses, and that has allowed us to get a good picture of what's actually happening in these cats. And it is very consistent. When we zoom into different areas of the genome of these viruses, when we're in ORF1b, it is all feline coronavirus, FIPV related, FCOV related, whether the repositories are quite poor and don't really tell you whether it's FECV or FIPV that the cats are presenting with, but the important thing is it's feline coronavirus. So the cypriot samples are green, cat samples are pink, dog samples are blue. When we look at the latter part of the genome, we're still closely related to the feline coronaviruses, all fair and square. But when we look at the spike protein, this is closely related to the canine coronaviruses and most closely related to what is called pantropic canine coronavirus. So what's happening? We can then run a bootstrap recombination analysis for the whole genomes that we have, and we see very reproducible, clear breakpoints. All the viruses that we have seen have exactly the same recombination points. So they recombine at ORF1b and at the near end of spike. So it's a beautiful kind of spike-only recombination, which we have seen similar actually to ancestral variants for SARS-CoV-2, as well as MERS coronavirus. However, very early on, we also saw, oops, there's an animation here missing. We saw a very interesting feature here, that in some of the spikes, we saw a deletion in spike as well, which raises more questions, and we'll get to that. But what is the closest relative to the spike protein that we've seen? It's a pantropic canine coronavirus that was first detected in Italy in 2005. And these outbreaks showed a higher pathology in the infected puppies primarily, and a very kind of systemic infection, just the way we actually observe it in the cats. This virus was traced all over Europe in the following years, but it never seemed to cause any major outbreaks beyond the initial outbreaks in the puppies. So luckily, that was hopefully a storm in the water glass. But we're now basically 20 years later, and suddenly we see this recombination into a cat virus. So as we highlighted, there has been the question of what actually makes FECV, the benign version of feline coronavirus, go to a feline infectious peritonitis virus. And one of the suggestions is the furin cleavage site between S1 and S2. Well, feline coronavirus serotype 2s and canine coronavirus serotype 2s don't have a furin cleavage site, so it cannot mutate. So it's not that. There are specific regions in the genome that could be associated with the mutations from FECV to FIPV. Nothing is obvious. And nothing has been previously described either. But we have this deletion in domain 0. When we look at the spike organization, we see the many different domains that form a functional spike protein. And interestingly, in most of our cats, this domain 0 is deleted. This is reminiscent of something. This is exactly what happened between transmissible gastroenteritis virus and porcine respiratory coronavirus. But porcine respiratory coronavirus is more benign. So why is this domain deletion suddenly potentially more dangerous? Porcine respiratory coronavirus has a preliminary infection in the upper respiratory tract. It also binds to aminopeptidase N, and TGV has an enteric binding phenotype. When we look at the spike protein, the short version loses these wee dangly bits, so to speak. And that could be part of an important change that could happen. Some of these dangly bits are normally associated with sialicide binding, which can be helpful and hindering when you're infecting cells. It can be retention on the surface, or it can help you grab onto hooks, let's say, to actually get into the cell. We then worked out that in the individual spikes that we see, the domain 0 deletion is basically cat-specific. So this must be an in-host deletion, because the rest of the viruses is pretty much still identical, but each individual cat has a beautiful domain 0 deletion. It's always in frame, it always makes a functional spike, but as you can see, very few of these, only the kind of green ones, have the same deletion, and that they're not correlating in a time or space continuum. And it's the case in over 90% of our FIP cases. So we worked with David Wieser's lab at the University of Washington, and Ale really quickly, actually, within the course of 2 months, managed to get the structures on these spike proteins. They look very similar to canine coronavirus structures that we've seen before, so there's nothing new there, which is also good news because we could pretty much predict it's definitely still aminopeptidase N, which is the receptor. But you can see here, when you look at these, you have the domain 0 kind of swinging out on the side, and it's reasonably flexible, and it can actually be in a swung out or in a proximal conformation. When you look at the accessibility of the receptor binding domain, once you lose that domain 0, it's very accessible. But it's still glycan shielded, which is quite good for immunogenic protection. When we look at the evolutionary structure, we're very much close to the feline coronavirus 2s, which we kind of knew. But the interesting bit is that none of the domains, domain 0, A or B, actually have any sialicide binding function. And looking at the structural similarities, canine coronavirus, here the one that jumped into humans, has sialicide binding, far more enteric related, but the FCOV23 doesn't have any of that function. So losing that function will not make any difference because it never bound to sialicides. Interestingly enough as well, once we look at the second cleavage site in the FCOV23 spike, so the S2' site that's just before the fusion peptide, which is obviously crucial to cell fusion or virus cell fusion, we see that basically all the physiological proteases that are distributed all over the body can actually cleave that site. It's only furin that cannot cleave that site, which is what we all expected. There's no furin cleavage site there. But it means you can have plasmin and XAPC1, which are both secreted into the blood and into the brain. You have catepsin L, which is anyways pretty universal. And then you have an enteric protease like trypsin. So you not only have a very universal receptor, potentially, but you also have a very universal protease that can activate fusion of your virus. And of course, we hypothesized that aminopeptidase N was the receptor, but we could confirm with pseudotyping that aminopeptidase N was indeed the receptor for this feline coronavirus. And more importantly, we could pinpoint it to a glycan in the human ANPEP, which shows why it's not binding to human aminopeptidase N. It's down here in between the loop 1 and loop 3 that there's a specific glycan in canine and feline ANPEP that allows the binding of the virus to the aminopeptidase N. If you mutate that location, it's R741T, so an arginine replaced by tryptophan, then suddenly you get binding and infection with feline coronavirus. And vice versa, if you remove that glycan binding site by mutating the tryptophan to an arginine, then you lose the binding of the feline coronavirus, of the feline APN basically completely. What was also very interesting and slightly scary is how well, especially the short version, binds to, for example, gallina, so chicken ANPEP. So the spread of this virus could be far beyond what we currently know. And together with the lab of Gary Whittaker at Cornell and Annette, we tested a lot of pseudoviruses on different cell lines. And this basically replicated what we knew. We can infect feline, we can infect canine, but not human, simian, porcine. We could also not infect chicken cells, but DF1s are fibroblasts. Fibroblasts are not supposed to express ANPEP, so I wouldn't have naturally expected any infection in those cell lines. So more work to be done on chickens, but let's hope they're not really spreading this virus just yet. What was the biggest surprise to us was when we looked at the fusogenicity of the short spike. And this is a simple transfection of the spike protein into cells that are susceptible to the infection. And we see cell-cell fusion within hours. Especially the FCWF-CU cells are really susceptible to infection. And actually, we had to start taking pictures of the short version at 4 hours post-transfection. So seriously, as soon as you start expressing this thing, it starts fusing the cells together. And if you look at 12 hours, basically your entire cell layer is ablated because they're all fused together. This makes us think that the short spike product is very important in pathogenicity, because if the virus can fuse cells so quickly, it can actually spread very rapidly, cause a lot of pathogenicity, and also induce the cytokine storms that we sometimes see in feline infectious peritonitis. So in summary, we have a fast-spreading, more pathogenic version of feline coronavirus that causes feline infectious peritonitis virus. It's indiscriminate of age, and whilst we thought that the second and third waves that we have observed yet would lower the average age of infection, we've not seen that yet. So it's not the new kittens that are getting infected. It is a recombinant between FIPV1 and the canine coronavirus, pantropic one. And we seem to see a direct transmission, at least of the benign version. We only have about 6% of our samples are fecal origin, and we could only get 2 sequences of those yet. They're both short, but that's the big gap that we're missing, and I'll come to that. We see a lot of neurological cases, and both the universal distribution of ANPEP as well as the use of plasmin could actually indicate why this virus can actually infect the brain. Because, well, as long as it can get there, it has susceptible cells there to infect. And we think that the domain 0 possibly is the reason, or the domain 0 deletion is possibly the reason why these cats develop FIP. We need more asymptomatic cases to confirm this, but the way it behaves in cell culture, it looks quite worrying. So we have discussed all of this and why it binds ANPEP, but there remains a bigger question about alpha coronaviruses, because we have seen this in TGV, we have seen this now in the feline coronaviruses, but it has also been observed in porcine epidemic diarrhea virus, that the virus can lose that domain 0. In pigs, it's always made the virus less pathogenic, but what happens if this happens again and again in different hosts? And why does this happen in host? What triggers that recombination? My hypothesis is that there's still a structural stability advantage to the full-length spike. So for the virus to survive in the environment, the long version will still persist. But in host, it seems like the short version has a massive advantage, because it's most of the cats with FIP that have this version. So we have a large body of work. We want to look more into and PEP and the real virus. And we want to see what happens in the cats that have the FCOV23, probably the long version, but are asymptomatic. Well, we hypothesize it's the long version. It could be the short version. So we have a program of epidemiology that is starting, sampling lots of cats, also the asymptomatic cats. We have diagnostics that are almost ready to be used that look at that SS interface. The treatment works, luckily, so the antivirals, remdesivir, GS, molnupiravir, they work very well in the cats, and actually the shorter treatments are getting more affordable. They're still expensive. And of course, we want to look more into the virology, the stability of this virus, and in the longer term, what does it mean for the larger family or for the larger class of alpha coronaviruses, that we can have these in-host deletions, because that is something new and slightly scary. So with that, I'd like to thank my team, all of our collaborators. You have seen their names pop up. You can read all of them here. But thank you, and let's have time for some questions and not hold you back from dinner.